Cercospora was established by Frasinus in 1863. It is a large genus of more than 3,800 species. The genus was first monographed by Chop in 1953. Cercospora species are saprophytic as well as parasitic. Most of the species are plant parasites causing leaf spot diseases. Cercospora mycelium is branched, intercellular, with haustoria entering the cells. Only asexual reproduction has been reported in this fungal group, and the conidia are the chief means of reproduction. Conidia are produced on conidiophores acrogenously. They germinate to form new mycelium. Fungi imperfecti, which form spores directly on the hypha or on the sporophores, which may be free or aggregated, but never in pycnidia or acervuli, are placed in the class hyphomycetes. Cercospora also belongs to hyphomycetes. Species of Cercospora are economically very important because most of them act as plant parasites. The name Cercospora is derived from a combination of Greek words kerkok, meaning tail, and sporos, meaning seed, thereby meaning the filiform conidia of the fungus. The teleomorph state is Mycosphaerella johanson, which is a genus that has been linked with at least 30 different coelomycetes or hyphomycetes anomorph genera. Crous and Braun in 2003 re-examined the Cercospora and presented a compilation of more than 3,000 names published in the genus Cercospora. They separated the cercosporoid genera mainly based on a combination of characters, namely structure of conidiogenous loci or scars and hilli, the presence and absence of pigmentation in conidiophores and conidia. Braun in 1993 also established generic separation of Cercospora on diverse criteria including ontogeny, pigmentation, and ornamentation of conidia, conidiophores, and Vidya-mitra. The character of conidial scars stressed by digiton is an unambiguous taxonomic criteria and has also been adopted by other mycologists in the classification of Cercospora and allied genera. Cercospora species are commonly associated with leaf spots, but they can also cause necrotic lesions in flowers, fruits, bracts, seeds, and pedicels. In addition, several species of this genus are also known to be hyperparasites of other plant pathogenic fungi and are employed as biocontrol agents of weeds. Now, general symptoms and damage of Cercospora on different hosts. Symptoms caused by Cercospora species are variable. Leaf spots may or may not be present. Leaf spots range from a faint discoloration on both the surfaces of leaf to well-defined and conspicuous leaf spots with colored borders. Often, effuse fruit bodies are visible on the lower leaf surface when no leaf spots are visible. When the disease reaches a certain stage, the leaf may curl, dry, and often droop from the plant. Many species of Cercospora also affect the blossoms, fruits, succulent petioles, and young stem. Spots may turn the entire leaf yellow or brown, after which it shrivels and dies. Species of Cercospora are economically very important because most of them act as plant parasites. Some common species are Cercospora calotropis on Calotropis procera, Cercospora hibiscus on lady's finger, Cercospora rosicola on rose, Cercospora personata and Cercospora arachidicola on groundnut, and Cercospora viticola on sugar beet. Tika disease of groundnut is widespread in India and is caused by Cercospora personata and Cercospora arachidicola. When this disease is in serious form, the loss to crop yield ranges from 22% to 70%. Carrot leaf blight is caused by Cercospora caroti. The mycelium of Cercosporous fungi is branched and septate. When the mycelium is grown in culture, it is hyaline initially and turns greenish brown at maturity. They can live as parasites or saprophytes. When the fungus infects the host, the hypha are cylindrical and branched. When they grow intercellularly, they form lobed hausteria. In case of Cercospora arachidicola, the mycelium is intracellular as well as intercellular and lacks haustorium. In case of Cercospora personata, mycelium is totally internal and intercellular, which sends branched haustoria into palisade and mesophyll cells. Before the mycelium switches over to reproductive phase, the hyphae accumulates at some places and becomes compacted to form a brown to black globular mass of hypha called stroma. From the stroma arise conidiophores. Conidiophores may be colorless, variously pigmented, and pigmentation is an important taxonomic feature. Conidiophores may be formed singly or arise from hypha as lateral branchlets, or they are arranged in loose or dense fascicles. Thus, vegetative mycelium gives rise to erect conidiophores which bear a single conidium at its tip or apex. Cercospora reproduces only asexually by conidia. In cercosporoid fungi, conidiogenesis is blastic, determinate, and show sympodial proliferation. Conidia may be single or they may be held in chains. Cercospora is characterized by long, cylindrical, obiclavate, multiseptate, hyaline, or brownish conidia. They are ash gray to light brown in color. Conidia have constant length-breadth ratio of 1:10 to 1:150. The first formed conidium is pushed to one side and a new conidium is formed at the apex. Conidia are blown by wind or raindrops. As the conidium separates from the conidiophore, it leaves a scar on the conidiophore. On germination, a conidium forms one or more germ tubes, which give rise to a new mycelium. Thus, life cycle is completed. Sexual methods are either absent or not reported yet. Perfect stage of some fungi are known in the genus Mycosphaerella. Two anomalous groups of Cercospora are found to exist, that is those with colored conidia and those with hyaline conidia. The conidia of Cercospora species are either straight or curved. They can be acicular, filiform, obiclavate, or may have a combination of shapes. The conidia of Cercospora are characterized by hyaline or pale olivaceous pigmentation and are euseptate, that is, septa are formed by all existing wall layers. Carrot leaf blight. Causative organism of this disease is Cercospora carroti. Symptoms. Cercospora leaf blight shows leaf spots which begin along the margins of the leaves and result in leaf curl. Spots inside the leaf edges are small, circular, and tan or gray to brown with a dead center. With the increase in number and size of lesions, the entire leaflet withers and dies. This reduces photosynthesis. The pathogen also produces lesions on the petioles and stems, characterized by dark brown borders and tan to gray centers. The lesions may merge and girdle the stem, causing leaves to die. Damage occurs to host because the pathogen causes severe blight on the leaves and petioles. Now, disease cycle of this pathogen. The pathogen survives in the seed and on diseased crop debris in the soil. The fungal pathogen produces spores that become airborne and are spread predominantly by wind. Moisture is essential for infection because spores require surface moisture and warm temperatures to germinate. Now, Tika disease of groundnut. This is also a serious disease. Causative organism of this disease is Cercospora arachidicola and Cercospora personatum. Early and late leaf spots are the most common and serious diseases of groundnut. Together, they can cause losses up to 50% in pod yield. The disease is also known by different names: Mycosphaerella leaf spots, Cercospora leaf spots, brown leaf spot, peanut cercosporosis, and Tika disease of groundnut. Leaf spots damage the plant by reducing the available photosynthetic area by lesion formation and by stimulating leaflet abscission. Early leaf spot is caused by Cercospora arachidicola. Leaf spots first appear on the upper surface of lower leaves as faint brown to black pinpoint dots. As the dots enlarge to become brown to dark brown circular spots, a yellow halo generally develops as a border around each spot. This disease is frequently seen early in the growing season. Spots with an irregular shape can also develop on leaf petioles and plant stem. It can lead to defoliation and reduce yield if not controlled by fungicide sprays. Leaves that fall on the soil surface may trigger epidemics of certain soil-borne diseases such as southern stem rot. Now, late leaf spot. This is caused by Cercospora personata. Like early leaf spot, this disease can also cause defoliation, reduced yield, and increased incidence of certain soil-borne diseases such as southern stem rot. As the name implies, late leaf spot is most prevalent during the late part of the growing season. Lesions caused by Cercospora personatum are usually smaller. Commonly, they show no yellow halo. They are more nearly circular and darker in color than those of Cercospora arachidicola. Now, disease cycle of this pathogen. The two pathogens grow differently in the host. In case of Cercospora arachidicola, the mycelium is intracellular as well as intercellular and lacks haustorium. In case of Cercospora personata, mycelium is totally internal and intercellular, which sends branched haustoria into palisade and mesophyll cells. In case of Cercospora arachidicola, conidiophores are olivaceous brown, simple, geniculate, continuous or septate. Conidia are mostly confined to the upper leaf surface. They are sparsely placed, haline or pale yellow, obculavate septate, with round to distinctly truncate base and subacute tips. Conidia are 30 to 108 microns long and 6 to 8 microns broad. They are formed by abstraction at the tips of conidiophores. In case of Cercospora personata, conidia are restricted to the lower surface and cushions of conidiophores are formed in concentric circles. Subcytomatal cavities are filled by hyphal aggregation to form cytomata. From the cytomata arise conidiophores which rupture the epidermis. Conidiophores are haline to dark brown, septate or not septate. Sometimes branched, straight or flexus and show distinct geniculate, that is, knee-like bends. They are 20 to 30 microns in diameter. These form single conidia acrogenously. Conidia are obculavate or cylindrical, light colored. Their length is 18 to 60 microns, while breadth is 6 to 11 microns. Early and late leaf support pathogens are soil-borne. Infection by Cercospora arachidicola normally precedes that of Cercospora personata, but both diseases may appear within 3 to 5 weeks after growing season. Conidia are produced directly from the mycelium in crop debris in the soil following early rains and when deposited on the leaves of young groundnut plants by rain splashes and wind, they initiate the disease cycle. Both fungi are capable of producing tremendous number of spores on infected plant parts. Spore production is favorable by high humidity. Visible spores develop 10 to 14 days after infection. New spores are produced in spores on infected leaves. These spores will subsequently infect plants and produce secondary infections. On germination, a conidium forms one or more germ tubes, which give rise to new mycelium. Thus, life cycle is completed. Conidia can also perennate in soil or shells of fruits for some time. Thus, they act as main inoculum and help in the perpetuation of the disease. Infection of new host occurs by direct penetration through cytomata of upper epidermis. Leaf support can increase rapidly under favorable conditions as several secondary cycles may occur per season. Abundant peanut residue in the fields where peanuts are cropped continuously often results in early and rapid development of leaf support. Gray leaf support disease of corn is caused by fungus Cercospora zeamadis. This disease occurs throughout the maize growing regions of the world where the growing season is characterized by high humidity and temperature between 22 and 30 degrees. Overcast cloudy days favor this disease. Now symptoms of this disease. Mature foliar lesions are rectangular, gray to tan in color, long and run parallel to the leaf veins. Under severe disease, lesions may coalesce and blight the entire leaf. Initially, tan-colored lesions are confined to leaf margins as the fungi are not able to penetrate sclerenchyma tissue in the leaf veins. Early symptoms of infection include pinpoint lesions surrounded by yellow halos, which elongate later and assume rectangular shape. Lodging may occur if stalks are also affected. When disease increases, entire leaves can be blighted and lesions can develop on comb sheaths also. Severe blighting not only causes premature death of leaves but also reduces the amount of photosynthate, that is sugars, required for air fill. Grains are not properly filled by sugars because photosynthetic sugar producing leaves are damaged. Lodging is also major cause of disease. Cercospora geomatis, like many other foliar fungal pathogens of corn, is a poor competitor in the soil and can survive only as long as infested corn debris is present. The fungi are host specific and overwinters as mycelium and stromata in infected maize plant debris left on the soil surface. Infested corn debris on the soil surface is the source of primary inoculum for the next corn crop. The fungus colonizing this debris produces conidia as early as May. In late spring, in response to warm temperature and high humidity, conidia begin to develop on residue. These are blown by wind and alight on the lower leaves of the plant. The first appearance of leaf spot and its continuous progress throughout the growing season are heavily dependent upon the weather conditions. Environmental conditions required for both types of leaf spots are warm temperature and long periods of high humidity or leaf wetness. Potential damage from leaf spot is greater where levels of humidity and rainfall are high. Frequent irrigation with small amounts of water can also create prolonged periods of high humidity and leaf wetness favorable for the infection.